In this demonstration, we present INGOT, an in silico network-driven combination therapy approach. State-of-the-art approaches generally focus on designing drug combinations by combining existing drugs and are limited by availability of suitable drugs. Moreover, these approaches generally focus only on achieving efficacy, which may lead to efficacious but toxic combinations. INGOT addresses these limitations. Given a disease-related signaling network, INGOT uses network analytics and heuristics to predict target combination that achieves the desired therapeutic effect with minimized off-target effects using an influence maximization-inspired approach. Drugs can then be designed for these targets. In this video, we will demonstrate 3 features of INGOT using a signaling network related to triple-negative breast cancer. These features are importing the network, visualizing the network, and predicting target combinations. First, we select the signaling network file, if it has not already been preloaded. INGOT currently accepts 2 types of network files. A topology file without kinetics annotation describing the interaction between molecular species, and a network data file with kinetics annotation typically in the SBML format. In our example, we shall use the topology file of a human signaling network containing over 6,000 nodes. Note that this network has been preloaded into INGOT for this demonstration. We shall now visualize the human signaling network by clicking on the Action tab in the menu and selecting the Visualize Preloaded Network option. A configuration screen will pop up for the user to select the network that they wish to visualize and the type of visualization required. In our example, the human signaling network allows 3 different view types, namely, entire view, integrated breast cancer view, and triple-negative breast cancer view. Further customization is allowed with the view extension option that will include molecular species that are X-hops from the selected view. For instance, triple-negative breast cancer view with 3 hops will generate a visualization of a signaling network with molecules relevant to triple-negative breast cancer and those that are 3 hops away. The view extension options include nodes near the boundary of the network, hence reducing the risk of missing out potential drug targets in subsequent analysis. Clicking on the Visualize Network button initiates the visualization process. The Visualize graph either displays fold change or mutation frequency of the nodes. Fold change is used to measure changes in gene expression level obtained from the omnibus dataset. It is derived from the ratio of the gene expression intensity of the tumor sample to that of the normal sample. Mutation frequency reports the percentage of samples in the cosmic database that carry mutation in that particular gene. For fold changes, nodes are represented in green if they are overexpressed, and in red if they are underexpressed. The node size indicates the extent of over or underexpression. The user can switch between from viewing fold change to mutation frequency by toggling the radio button. For mutation frequency, the size of the node indicates the frequency of the mutation. Larger-sized nodes are mutated more frequently. Finally, to predict target combinations, click on the Action tab and select Compute Target Combinations. A screen will pop up and guide the user through the different steps. First, select the disease nodes related to the signaling network. Disease nodes are molecular species that are implicated in the disease, and regulating them may restore the physiological state. For example, SEMA5A, a pro-angiogenic gene, typically exhibits high expression level in triple-negative breast cancer. The disease nodes can either be selected from a list by the expert user or generated using rules. For instance, setting disease nodes as those having expression fold change more than or equals to 2.33 and with mutation frequency percentage more than or equals to 2 selects DST, SEMA5A and CDKN2A as the disease node set. In the next step, we select the off-target rules. These rules are for computing an off-target score, which is used to rank different target combinations. A lower off-target score implies a combination with potentially lower toxicity. In this video, INGOT seeks combinations that minimize perturbation of essential nodes and nodes with high weighted out degree. Nodes with high weighted out degree are more likely to propagate the perturbation. We then set the combination size as 3 and use the greedy approach with subgraph heuristic to compute the target combinations. This heuristic partitions the network into subgraphs, each containing one or more disease nodes, then for each subgraph, identifies suitable target to add to the combination. Target selection depends on the disease node cover and the off-target score of the resulting combination. Clicking on the Compute button initiates computation of target combinations. After computing the target combinations, the target combination nodes are highlighted in green and the disease nodes in red. The Disease Node List tab displays a summary of the disease nodes, their fold changes, and mutation frequencies. A literature survey of these disease nodes reveals that they are implicated in metastasis and invasion, which is likely to be a high-priority cancer hallmark for triple-negative breast cancer. The user can also select the View option menu to visualize the disease nodes. We provide two view options: View Node Neighborhood and View Disease Node Connections. The user has to select the disease nodes the viewer required and then click on the Visualize Disease Node button. The View Node Neighborhood option displays the one-hop neighborhood of the selected disease nodes. User can zoom into specific disease node by selecting it, and the relevant nodes will be highlighted. The View Disease Node Connections display all nodes in the pathway connecting the selected disease nodes. User can view the nodes between particular disease node pair by selecting them from a list, and these nodes will be highlighted. Finally, the Target Combination tab at the bottom left panel displays a summary of the best predicted target combination. This combination consists of SEMA5A, TP63 and SMAD3. Each of these components has been implicated in cancer by different research groups. For instance, TP63, a p53 family member, controls a survival pathway that directly mediates cisplatin sensitivity in triple-negative breast cancer. Hence, the predicted combination is likely to be biologically relevant. Note, however, that further wet lab experiments are required to verify the predicted combination. We provide similar visualization options for the predicted target combinations. User can select a combination from a list. Note that the list is ranked according to the off-target score shown in square brackets. The visualization can be done together with the disease nodes by selecting the Include Disease Node in View checkbox. Note that the newly selected target combination will be visualized on the graph panel. In summary, INGOT uses a systems biology-based approach to predict target combinations and is useful in helping scientists analyze disease-related signaling network and to formulate hypotheses for guiding the design of further wet lab experiments.